Following the theme currently, this project was about using the BV-BRC MCP server to execute a bioinformatics workflow in a single chat session. And thanks to my team for helping get to this point and develop the server to something that we can actually use to conduct a bioinformatics analysis. So the example that we used was based off of a existing workshop example that Rebecca has presented at some of our workshops. This is the ideal workflow, but the steps that we're able to do in this analysis was the metagenomic binning and the taxonomic classification. So limited in the entire workflow, but an important first step in making this something that can happen end to end. For this analysis, we also included 2 publications for reference. We supplied the titles to these publications in the chat session, and ChatGPT went out, examined the text in those publications, and incorporated some of their insights and genes into the analysis, which you'll see in certain steps. For this presentation, I have a series of ChatGPT screenshots. This first one is the initial prompt. We said, start a new analysis, gave it the SRA sample ID along with one of the publications to sort of kickstart this. We gave it the goals that we wanted. We wanted to know what organisms were in this metagenomic sample, extract those genomes, and attempt to predict any antimicrobial profiles. We asked it to also describe the workflow in detail, and then using the MCP, launch the analysis jobs that would be used, and so we can analyze those outputs. So this is a screenshot of the workflow description. It took our prompt. A couple of steps were done before this point, but this is a summary of what it believes the end-to-end workflow should be for this sample. So it lists out we need to do taxonomic classification, slightly redundantly metagenomic binning, and then suggests using the publication. We did not suggest anything about macrolides or azithromycin, but it went out looking at the publication, identified those as the analysis goals of that publication, and incorporated that into our own workflow, sort of showing how we can have the tool server, but another very powerful aspect of using these models is to incorporate existing literature and enhancing your own analysis. This is a screenshot of the metagenomic binning job submission. The tool had, using our initial prompt and the MCP server parameters, constructed this to submit a job to our analysis servers. So one thing to note is that these are not in-time jobs, so we have to query our jobs database to see if it is in progress or if it's completed or if there has been an error. This first screenshot at the top shows the response when the job is still in progress. So, it looked up the job, saw it was in progress, told it to pause, came back later and asked it to continue if it had been completed. And as you can see, we get past that first step and it's immediately trying to list out the files that were output in the successful job. Here's it listing it out and describing the information from the metagenomic binning job. And then from those files, we then went to the next part of the analysis, which was to specifically look at the resistance and susceptible genomes for azithromycin using the organism that had been identified in the metagenomic sample from the metagenomic binning step. Slightly cheating here, we did have a publication that used the same organism. Let's see. In the previous step, as we can see at the very bottom, it is telling us that with high confidence, we've identified that the only major organism in this metagenomic sample is the Streptococcus pneumoniae. From there, what we want to do after identifying the organism is to collect all of the existing genomic information that we have for those similar organisms. And so what I'm showing here in these screenshots is we have a tool to download information from our data API. So here we are looking for all of the data in our genome AMR collection for the organism Streptococcus pneumonia, and specifically susceptibility to this particular antibiotic. And using that selection of genomes, we create a genome group, which is a data object that is very important in the BV-BRC analysis services. We then used those genome groups to ask questions about the particular genes that were identified in the reference studies. And so here we went out and found the resistance susceptibility profiles for the list of genomes in that genome group. And ChatGPT made a nice summarized table of those results for a small set of genomes in that set. While this was a great first step showing that we can, in a single chat session, execute these bioinformatic workflows to help us do science, some things to improve. We can have the better incorporation of different publications, maybe provide, like Galaxy has been showing, some workflows in order to give it a better idea of how to conduct this analysis based on prior publications and analyses. We can improve our own MCP server. And one of the bigger steps is, I guess, to make this sort of analysis an asynchronous chat so that you do not have to prompt it to continue. You have an agent or something that can sort of monitor it as it goes, make the necessary adjustments. You walk away for 3 hours and hopefully you have a nice set of analysis results to look at. So that was, I think, great progress for us. Any questions? All right, thank you.